In the 17th century, Dutch naturalist Antony van Leeuwenhoek used a microscope to peer into the world of cells, saw bacteria, erythrocytes, and spermatozoa, and laid the foundation for microbiology. In the middle of the 19th century, John Lawrence Smith put forward the design of the inverted microscope. Now scientists were able to observe living cells directly within their culture dishes. But living cells are semi-transparent and almost indistinguishable in a bright field. In 1930, Fritz Zernike developed phase contrast, which made it possible to visualize transparent objects. This was achieved without the need for staining or fixation. Working with transparent cultures became significantly more effective. And in 1975, Robert Hoffman introduced Hoffman modulation contrast, which is yet another method for visualizing living objects. Good day, I am Marina Lobach, the Scientific Director for the MAGUS project. Today, I will elaborate on how the MAGUS V360 inverted microscope integrates these diverse research methodologies into a single, cohesive system, thus facilitating precise and comfortable observation of cell cultures. For this review, we didn't use any expensive reagents or rare samples. All photos and videos taken by us on the MAGUS V360 are from available materials. Even with such simple examples, the instrument demonstrates a level of quality suitable for serious scientific research. If you work with cell cultures, living tissues, or within the field of biotechnology, then do stay with us. The MAGUS V360 is truly capable of pleasantly surprising you. It's a versatile instrument for medicine, biology, pharmacology, and virology. With it, you can examine both stained and unstained objects directly in culture dishes. The inverted microscope design accommodates various types of culture dishes, including Petri dishes, multi-well plates, roller bottles, and other containers up to 75 millimeters in height. Without the condenser, it can accommodate labware up to 187 millimeters high. The MAGUS V360 supports 3 methods of contrast, even in its basic configuration: bright field, phase contrast, and emboss contrast. The 4th method, Hoffman modulation contrast, is optional. The V360 has a well-designed ergonomic structure. This model offers comfort even during many hours of work with the microscope. The binocular head with its 45-degree tilt reduces strain on the neck and back. By rotating the tube, you can adjust the interpupillary distance to suit the user's height. The digital camera is installed in the side visualization channel, which provides an unobstructed view of the specimen stage. The light path switching knob has 2 positions. The light either goes completely into the eyepieces, or it is redirected to the camera. This method prevents unwanted light artifacts in your photos. The microscope kit includes 2 C-mount adapters, 0.5x and 1x. The selection depends on the camera's sensor size and the specific tasks you need to accomplish. The focusing knobs and the specimen translation mechanism are positioned at a low height, allowing your hands to rest comfortably on the table. The mechanism's movement is both smooth and requires minimal effort. The ring on the right side regulates the stiffness of the coarse focus movement. The universal holder for culture dishes reliably secures various types and sizes of culture vessels. When working with a culture bottle, a metal insert with a specialized opening is provided. This feature allows the objective lens to safely clear the edge, thereby preventing any potential damage to the optical components. The glass insert simplifies the handling of wide dishes and allows you to move them freely by hand. A complete set of holders for various sizes of culture dishes will significantly enhance user comfort. Collectively, these features enable prolonged use of the microscope without fatigue, ensuring peak efficiency. Next, let's explore the optics and the revolving nosepiece. The revolving nosepiece is coded and holds 5 objectives. The standard configuration includes 60mm parfocal plan achromatic objectives with a long working distance, designed for a dish bottom thickness of 1.2 mm. Plan achromatic objectives ensure a sharp, uniform image throughout the field of view. The set of objectives includes 4x, 10x, 20x, and 40x magnifications. The 10x, 20x, and 40x objectives are for phase contrast, but work for bright field and emboss contrast too. Let's take a more detailed look at the intelligent brightness control system. The MAGUS V360 memorizes the brightness level selected by the user for each objective, and it automatically sets this level when the revolver is turned. This saves time and significantly enhances user comfort. The illumination is provided by a 3-watt LED that has a lifespan of 50,000 hours. The LED's color temperature doesn't change when you adjust the brightness. This is important for photography. The LCD screen displays the operating parameters, objective, brightness, and sleep or power-saving modes. The settings are adjusted using a single knob, making it quick and intuitive. Now let's delve deeper into the various contrast methods. Bright field is the classic method, suitable for high-contrast specimens. Phase contrast is used for transparent structures like membranes, nuclei, and vacuoles. This means no fixing and no staining are needed, both of which can damage the cells. The slider contains a single insert that is compatible with 10x, 20x, and 40x objectives. This saves time. There's no need to switch the slider when changing objectives. Emboss contrast. This is an accessible method for creating a 3D relief effect, which adds shadows and helps to visualize the specimen's structure more clearly. It doesn't require additional optics. Standard objectives and 2 sliders are sufficient. This method generates a pseudo-3D effect, eliminating the glare and halo artifacts that typically occur on thicker specimens when using phase contrast. The 3D effect is achieved through special processing of the light path. A slider with a sector diaphragm is installed in the condenser, and one with a set of masks in the visual attachment. Proper adjustment creates an oblique illumination effect. Emboss is versatile and practical. It is compatible with both glass and plastic culture dishes. Hoffman modulation contrast is an optional technique that offers excellent detail without halos or artifacts, making it effective for highlighting the distinctions between different structures. It is effective for studying the real-time dynamics of living cells. Hoffman objectives are outfitted with an amplitude spatial filter, a modulator featuring 3 distinct zones: dark, gray, and transparent. It doesn't change the phase of light, but only regulates its intensity. This entirely eliminates halo effects, providing a clear depiction of object boundaries without any distortions. The Hoffman condenser is installed in place of the standard one. Its design includes an aperture diaphragm and a rotating polarizer. The slider includes 3 inserts for 10x, 20x, and 40x objectives. For both fundamental and applied scientific research applications, a phase contrast kit is offered, featuring 4x, 10x, 20x, 40x, and 60x planned semi-apochromatic objectives. The 60x objective features a correction collar for use with culture dishes that have varying bottom thicknesses. These objectives provide a high degree of aberration correction and produce a clean, bright, and high-contrast image. Select the research method best suited for your specific task. Phase contrast is particularly effective when working with transparent specimens. However, it can sometimes introduce visual noise when observing thicker objects, as it registers phase shifts across their entire volume. The Hoffman method is sensitive exclusively to the optical density gradients found within the focal plane. This provides an optical section, offering a clear visualization of the chosen layer without interference from structures located either above or below it. The method is suitable for observing real-time dynamics, cell division, and organelle movement, allowing for the visualization of fine details in living samples with high contrast and without artifacts. So let's summarize. The MAGUS Bio V360 inverted microscope is perfect for medical, virology, biology, and pharmacology labs. This microscope proves highly effective for handling various cell cultures, meticulously observing dynamic cellular processes, and thoroughly studying how cells react to different drugs. The basic package includes everything essential for operation: the microscope with its objectives, a visual head equipped with eyepieces, an illuminator, a condenser, phase and relief contrast sliders, and a versatile culture dish holder. Additional modules broaden the scope of tasks that the Bio V360 is capable of addressing. Eyepieces with different magnifications provide a flexible range of magnifications. The phase contrast device with planned semi-apochromatic objectives is ideal for scientific research applications. Hoffman modulation contrast provides detailed visualization of the tiniest details in living samples, with excellent contrast and without any halos. A digital camera transforms the microscope into a comprehensive system for photographic and video demonstrations. The software that comes with the camera will enable you to accurately measure various objects. Accessories for various shapes of culture dishes will increase your working comfort. The MAGUS Bio V360 microscope combines intelligent illumination, excellent optics, and well-designed ergonomics, making it simple to operate while being fully equipped for research tasks. You can find detailed specifications for the model on the MAGUS Micro website. Want to test the microscope? Please come to our demo hall, bring your samples, and we will work together to select the best configuration for your specific task. See you soon, colleagues.